All right, let's take a look at carbon. Carbon is hugely important for biology. It's the basis of all life forms that we know of anyways. And we'll look at why that is, and then we'll look at some of the reasons and the ways in which it is able to do so many things. That'll come up with the functional groups, as well as we'll look at a couple of reactions that happen with certain compounds that we'll be talking about in more detail. So carbon itself as an atom, we know atomic number 6, and so it's got 2 electrons on the inside, it's got 4 electrons, valence electrons on the outside, and what this means is that it is able to, it wants to have 8 on the shell there, so it was able to share with 4 other atoms. So you can end up forming 4 covalent bonds. The reason why that's important is because if you want to have self-replicating entities, living things, they have to be complex. And if you can connect a carbon to 4 other things, so there's a carbon in the middle and you can connect it to 1, 2, 3, 4, then you can form some ridiculously complex structures. If carbon could only form 1 bond, then it could only ever maybe form a chain of like a bunch of carbons, which is great, but not particularly complex. So because of the way in which carbon can form so many different bonds, it can form 4 individual bonds, but it can also start forming double bonds and triple bonds as well, and then you can get things like branches coming off of substances and all sorts of complexity with it because of the number of bonds it's able to form. So this is the reason why carbon is the backbone of living things. It's also small enough that these bonds are able to be quite strong. If you start doing something, if you look at the periodic table and say, oh well, what about silicon? Why can't we have silicon-based life forms? And I don't know, maybe we can, but we haven't found any yet. And it's probably because when you start getting those larger atoms, it can form these bonds, it can share these electrons, but they end up being further away from the nucleus, so they're not as strongly held. So carbon can form 4 bonds, and they're shared bonds, not just ionic bonds, so they actually share bonds, so you get the complexity there, and is able to hold on to those bonds. So they're relatively strong. The other elements that we're going to have here that we're going to see a lot of are going to be hydrogen, oxygen, nitrogen, sulfur, and phosphorus. So those, along with carbon, make up the bulk of most molecules that are involved in biology. Quick review of some of the prefixes. So again, if we want to talk about the number of carbons, maybe an entire molecule, or maybe just in like a part of a molecule, we have these particular prefixes that we use to do that. If you can memorize up to bute, the rest I'm sure you'll recognize from your general knowledge or your shapes or math or somewhere in there, or octopi. All right, variation in carbon skeletons contribute to the diversity. So as I was mentioning earlier, the fact that you can change the length of them, so you can put them all in a row, which is great, but you can also have different sized rows, and so you could have 2, or you could have 3, or 4, or 5, or so on and so on. So they can actually form different size molecules, which is great. You can't do that if you have something like, I don't know, just hydrogen, right? Like if you just had a hydrogen to a hydrogen, that's as far as you're going to get. You don't get a chain at all. Plus, these chains can be branched because, again, carbon can form 4 bonds. So let's say, I don't know, maybe you could have oxygen, which can form 2 bonds. Maybe you can imagine there's some sort of crazy oxygen chain you could make. Wouldn't be very stable, but maybe possible. But it would only ever be linear. Carbon, however, because it can form more than 2 bonds, you can actually start forming branches, and not just 1 branch, but you can actually form 2 branches off the same carbon. Plus, you can form double bonds and triple bonds, not here, but you can actually form triple bonds as well. So that gives you more variety, and you can start getting these chains. So imagine these chains, if you get long enough, they could actually connect back to themselves, and you end up with these ring structures. Plus, you can put all these things together, and you get super complex molecules. So one of the ways to add complexity to a molecule, we looked at those, the fact that they can form different length chains, they can form branches, they can form double bonds, they can form rings. That's all great, but then you can start throwing in other atoms as well, and ways in which it's arranged with these other atoms. When we have this cluster of atoms that are attached to a molecule, so this isn't just by themselves, but this is a part of a molecule, we call this a functional group. So let's take a look at a few of them. Looking at some of the effects of these, here's look at these 2 molecules here. So we got this guy and we got this guy here. Pretty similar in terms of the shapes of them. So each one of these is a carbon. We tend to not always draw the carbons, but each time those lines intersect, those are carbons. And so the backbone of these two molecules looks pretty similar, but if you look, you can see the two differences. They're outlined. This one here has a double bonded oxygen, and instead this one has an oxygen with a hydrogen in it. This one here has a carbon sticking off of it, and you don't see that on the top molecule. So there's subtle differences between these two molecules. The point is, if you take a molecule and you make a small change to it, you can have a hugely different biological impact. And so these two particular molecules, you recognize the name of at least one of them, these are hormones, and they are relatively similar to each other. They're molecularly quite similar, but they have a slightly different change in them, a slightly different functional group. So you have an OH versus a double bonded O, and this one here has a methyl functional group on it, and there is none on this one. So not that big of changes. However, it gives you the difference between a hormone that males will use for certain secondary sex characteristics versus a hormone that is more common within females. And so you can see this estradiol versus testosterone can have huge biological implications with only very small molecular changes. So let's look at hydroxyl as the first functional group, OH. This is not to be confused with a hydroxide ion. So we mentioned with acids and bases that you can have a hydroxide ion, and this is a hydroxyl group. So they look terribly similar, but this is hydroxide, and hydroxide is not hydroxyl. Hydroxyl, the line, when we draw it, so look, there it is right there. That line is showing you that it's bonded to a carbon. When you draw a hydroxide ion, this line that we draw is an ionic charge. It's an ion. Two very, very different things. So don't confuse hydroxyl with hydroxide. We're going to be talking about the functional group, the bonded thing to something else. It is a functional group. It is part of a larger molecule, not an ion. So hydroxyl, OH, sometimes referred to as an alcohol functional group, because it's what you find in alcohols. Lots and lots of different types of alcohols, and they have hydroxyl groups to them. Just because it has a hydroxyl group doesn't mean it's an alcohol, but if it is an alcohol, it does have a hydroxyl group. Some of the characteristics of them for this one is it's really polar. You've got oxygen bonded to hydrogen, therefore you have a strong dipole, therefore you have large partial charges, and you have the opportunity for hydrogen bondings to form. So this will make molecules quite polar. This is the carbonyl functional group, and it is an oxygen double bonded to a carbon. So it doesn't have a hydrogen of its own, it is just actually double bonded to that carbon. And so you can have a chain of carbons, and if one of them has this double bonded O sticking off of it, that is a carbonyl functional group. It does add some polarity to it. You find it with ketones and aldehydes in organic chem. We find it in things like sugars. We'll see this functional group in. Then there is the, not to be confused with the carbonyl, this is the carboxyl functional group. And think, I don't know, it's got the carbon, double bonded oxygen, and the OH. So it's kind of like a alcohol hydroxyl and a carbonyl together. Now they have to both be on the same carbon for this to work. And if they're both on the same carbon, the double bonded oxygen and the OH, then we have a carboxyl functional group. Really important for acidic substances. So this actually leads to the molecule it's on having some acid properties. It can actually lose its hydrogen, and that makes it an acid. And so here's a picture of this is vinegar. We have a molecule, and it happens to have this carboxyl functional group on it. It can lose its hydrogen, therefore it's an acid. That's what vinegar does. Also important for biological systems. So we have these molecules that will have this functional group on it that are able to act as acids. Then there's the amino functional group, and you probably know where this is going to be found. Amino acids have it, and take a look at that. There's the amino one we're talking about now, and look at that. There's the carboxyl one that we were just looking at. So this is why it's called an amino acid. There are lots of different types of amino acids, but they have an amino functional group, and they have an acid functional group, and then they have something else that makes them unique to their particular own amino acid. And we have a phosphate. So we'll talk about this when we're doing our metabolism, but it's basically, it's kind of like a phosphate ion that is actually attached molecularly to something else. So it is got 4 oxygens and 1 phosphorus on there. Methyl is really just a carbon, and it's a single carbon, so that's why it's a methyl functional group. If it was 2 carbons, it would be an ethyl functional group. If it was 3, it would be a propyl functional group. But methyl is quite common, so just a single carbon sticking off of another molecule. All right, so here are the main ones here. Again, a hydroxyl, carbonyl, carboxyl, amino, and then we'll see some phosphate functional groups throughout the course as well. Quite common, and again, not limited to, because we'll see some variations in here, but carbohydrates definitely have hydroxyl groups to them. Lipids definitely have these carbonyl groups on them. Proteins have carboxyl groups on them, as well as the amino group on there. And then again, with energy and when we start talking about nucleic acids, we're going to see these phosphate functional groups. Quickly looking at, we're not doing a course on biochemistry entirely here, so we're just going to go over this fairly briefly. We're going to look at acid-base reactions. Not much, but a tiny bit, pretty much just the definition of an oxidation reduction reaction. And then this third one, a little bit more important to what we're focusing on, but a condensation / dehydration, and the reverse of that, which is called hydrolysis. So first one, as we mentioned when we were talking about acids and bases, if you combine an acid and a base, they're able to neutralize each other. And I mean, it doesn't have to be a complete neutralization, but they will react with each other and produce water. The acid produces the hydrogen, the base produces a hydroxide in some way, and that hydrogen and hydroxide come together, they give you water. May also produce a salt, but not always. It will always give you water. Buffers, again, as we mentioned, will resist a acid-base change. So if you don't want to have a solution's acidity change too much, you can add a buffer to it. Oxidation reactions involve electrons. And so if you move electrons from one thing to another, it's an oxidation reaction. And when we're lazy, we just call them redox reactions, the reduction and oxidation. Loss of an electron is called oxidation. So the thing, if something loses electrons, we say it's been oxidized, it's gone through oxidation. When something gains electrons, we call it reduction, and we say it's been reduced. A little bit counterintuitive there. The gaining of electrons is reduction. So to remember that, I would recommend this particular way of doing it. Oxidation is loss, reduction is gain. Oil rig. So if you have A here, and it's got a bunch of electrons, and it becomes something that doesn't have those electrons, notice they're not there anymore, because they have actually gone over to B. B didn't have any electrons, and there it is, it's got some now. And so A has gone through oxidation. B has gone through reduction. Oxidation is the loss of electrons. Reduction is the gain of electrons. Last, dehydration and hydrolysis. So several types of macromolecules. We're going to spend a lot of time talking about those, but these are big, really important molecules. I'll look at a few types of them. They're going to be built. They're big, so they're going to be built from smaller pieces. So they get assembled through these condensation or dehydration reactions. And you notice those two terms have something similar. It is that you're talking about water with this. And so if you take these monomers, you can stick them together through a dehydration or condensation reaction. In this, you've got these two pieces you're going to be putting together, these two monomers. One of them is going to lose an H, the other one's going to lose an OH, and you put those two together, you get water. That's why it's called dehydration. You get water out of it. And when you do that, these two things, the one that lost the hydrogen, the one that lost hydroxide, sorry, I said hydroxide, not hydroxide, the one that lost the OH has lost that functional group, and now it is going to be joined together. And so you're going to build these larger molecules from these smaller monomer components. So this is dehydration. Synthesis is the term we're going to use to describe it. The opposite of that, if you had something that was big and you wanted to split it apart you can do that using water. So hydrolysis, the splitting of something using water, is a way that we can break apart macromolecules. You eat a bunch of protein and you want to use those amino acids to build your own proteins. You can break those proteins apart, those really big molecules. You can break them down into their amino acids by doing a hydrolysis reaction. So we have these two processes that we can do. We refer to the breaking down of larger molecules as catabolic, and then the reactions that combine simple molecules into larger ones, the sort of building up of them, anabolic reactions. So we're going to look at those two when we get into metabolism. So let's say we had this molecule, it's got a great name, and this molecule, which is the same thing. So these are two monomers. And what we can do is we can actually, get rid of that, we can actually join them together, plus. So we can put them together by taking the hydroxyl functional group and the hydrogen and making water out of them. So the OH and the H go together. But now you'll notice the oxygen here would be missing a bond, right? Because it was bonded with hydrogen, you took its hydrogen. And right here is a carbon, and now it's missing this hydroxide. So what's going to happen is that carbon there and this oxygen here are going to get bonded together. So you see that the oxygen reached over and bonded with that carbon. That's what this line is showing here. And so these two monomers are now connected together. So you can start building larger molecules with it. Fats can do it too. You may have heard of triglycerides or fatty acids. These are names for different parts of fats here, but you can build fats by taking glycerol and fatty acids, and again, creating water, doing dehydration synthesis. If you take the OH from one, the H from the fatty acids, you take them out, you would get H2O, and now that carbon is going to be directly bonded to the oxygen that was at the end of the fatty acid that lost its hydrogen. Now it's going to bond to something and it'll bond to that carbon, and you end up with these guys stuck together. This can happen 3 times with glycerol, making a triglyceride. Proteins can do it too. Again, we mentioned they're made of amino acids. Same thing. Take a hydroxyl group from one, a hydrogen from the other monomer, put them together, and you start building a chain. You can build a really big complex protein and make life. Nucleic acids, last one, and we'll go over these in more detail, but same thing. Dehydration synthesis, connecting monomers together to make larger molecules.